Trypan blue? What does it do? It enters dead, but not living, cells and shows them to you. It also, fun fact, kills trypanosomes and hence the name. But we're more typically using it in the cell counting game. So Trypan blue stain is a stain that we can use to look and see what proportion of cells are viable. So which of them are like alive and which of them are dead. And it's really useful if we're trying to do cell counting, where basically count how many cells are in a solution so we know how to like plate them and things like this. We want to make sure that we're only counting the live cells, and so we can pre-stain with this Trypan blue. Cells that are dead are going to take in this stain because their membranes are all damaged, and so then the stain can get in. But if the cells are healthy and their membranes all intact, then this dye can't get in, and so it's excluded. And so this dye exclusion method is able to then stain the dead cells blue. The living cells will remain non-stained, at least in the beginning. That's why you have to do it quickly in like the first 5 minutes or whatever. You have to actually look for it. I'll tell you more about this nitty-gritty details and stuff later. But this is going to then allow you to see which proportion are alive, which is helpful for the counting, as well as being able to see just like, how are my cells doing? Was I too harsh with them when I did that thing? Are their growing conditions okay? Things like that. And so it's really, really useful for that. So I'll also talk about some artifacts to watch out for, things like that, like undercounting, overcounting, things like that. Some alternatives. So typically, like, there's other alternatives like fluorescence and things like this that are more commonly used for determining the viability when we're doing when people are doing like FACS or various cell sorting based things. But when it comes to in the lab, in the tissue culture room, when you're just trying to see how the cells are doing, trying to count them, Trypan blue is very, very commonly used. It's quick, easy, just need a normal microscope, and so here's how it works. Also the trypanosome stuff, because it's really cool. So chemically speaking, Trypan blue is an azo dye. So it's called an azo dye because it has these azo groups, and it's a dye because it's colored. And the reason why it's colored is you see all these rings. It has this extensive conjugation or resonance, just delocalization. Basically, it has these what we call aromatic rings, which, long story short, they allow it to absorb visible light. And if you absorb visible light, then something looks colored. So white light has all the colors of the rainbow, and so if something's absorbing, it's stealing one of those wavelengths, you're going to see other wavelengths. The resulting is going to be different. And so this is going to be, in this case, it's going to appear blue. So we have this blue dye. And if this dye gets into cells, then the cells will look blue because the dye is blue. But the problem is, or I guess not the problem, is that the dye can't get into cells unless those cells are damaged. Cells are surrounded by this phospholipid membrane. So this double layer of these lipids. So you might think lipids, those are all greasy and oily and lipidy. Well, yeah, but on the surface of these lipids because they're phospholipids, they have these negatively charged groups on their head. And so this makes the surface of the cell negatively charged. Another thing about this dye is that it's anionic. It has these negatively charged groups. You often see it in its salt form. So typically there'll be like an Na plus here, and it'll be neutralizing this. So a salt is when you have a positive thing and a negative thing, and then they come together and they're neutral. But basically they're just hanging out next to each other. They're not actually like strongly bound together or anything like that. But this is going to be negatively charged, and opposite or like charges repel each other. So this dye is going to get repelled from the surface of the cells. It's not going to get in. And however, if there are like a damaged membrane, then there are holes in the membrane, or the membrane's just like, I don't know, totally exploded or something, then it can just diffuse in. So diffusion is just like the random movement of molecules. And so here there's no path in, it can't get in. But here, if you have these pores or you have some sort of damage to the membrane, now that dye can start to sneak in. And when that dye sneaks in, the cell is going to start getting blue. And then you can look under a microscope and see which cells are blue and which cells are not blue. And importantly, you can kind of like determine what's the like percent of blue to not blue? What's the viability of these cells? This helps you examine the health of your cell cultures. So, like, was the treatment toxic to the cells? Are these growth conditions okay? Um, Things like that. So if you're doing some sort of experiment, maybe you want to see, okay, is the chemical I added to test, was it toxic to the cells? And a further hint that it's the chemical itself that's doing things is if your carrier only control, so like your DMSO carrier, we talked about DMSO the other day, it's basically a solvent that helps you get compounds into cells when those compounds aren't very water soluble. And so if that carrier alone, if the thing that your compound was dissolved in, if those cells are doing fine, but your cells that you treated are dying, then it's probably something toxic. But you're probably going to want to follow it. You're going to want to follow up because there are lots and lots of different things that could cause the cells to be dying. These are just a few of the things that could be causing it. But anyway, it's a warning sign if you see this happening. If you see cells dying, and you want to know. Okay, so it's often though, like one of the most common things we use it for is actually when we're counting cells. So when we're growing cells in cell culture, we're often doing this in an adherent cell culture, or we can also do suspension cell culture. Adherent cell culture, basically you're growing the cells in a dish, and so or a flask or something like that, but these cells are stuck on the surface and they're going to form this like monolayer, just like a single layer of cells. And they're going to run out of room because these cells are going to keep dividing and dividing and keep growing and growing. And so we need to get them off of the dish and split them up and plate them in new dishes with more space. So we like dilute them out. We need to know how much space to give them. We need to know like the seating density. So basically your cells are going to get like overgrown on one plate, or you don't want them to get overgrown because then you'll start seeing a lot of that blue because your cells will start dying. Often we use trypsin to basically cut it off of the dish. And then we need to calculate how many cells there were so we can calculate how many cells to plate per dish in order to give them enough space and enough food, but also we don't want to just like plate a single cell per dish because these cells need to have like communicate with one another through these like chemical signals that tell them, okay, you can take down root, grow, do all these cool things. So you need to have a specific number, well, there's like a range of seeding densities you could do, but basically you seed the new plate with a smaller amount of cells than were in the plate that was overgrowing so that these cells have plenty of room, but also plenty of friends. And so you need to be able to measure how many the concentration of your cells. But you want to only measure the concentration of those live cells because those dead cells aren't going to contribute to anything. You want to know if there's too many dead cells, there might be some sort of issue and your cells might not even like survive if you try to plate them. It might be better to like go back to a new stock or something like that. But you want to know how many of the live ones there are, the viable cells there are. Typically, we measure this like in per milliliter, and then we can calculate how much to plate on a new dish. And so I'll get more into that details in a second. But how do we do this counting? So after we trypsinize the cells, or if you're doing a suspension culture, you basically just take a aliquot of the culture, or maybe you spin it down first, remove the old media, suspend it in less media. There are different ways to do this, but basically you just need to get a sample of the cells in a solution. So these cells, when they're stuck on a dish, we have to cut them off of the dish, and then basically what we do is then various things that we have to do to stop it getting cut off, so stop that trypsin that was cutting it off of the dish before the trypsin goes wild and starts cutting and killing our cells. But we basically, once it's cut off, then we inhibit it, typically by adding media with serum in it, which is going to inhibit the trypsin, which is the protease that's cutting them off of the dish. And then once we've inhibited it, then we spin it down and we remove the trypsin and everything like that, and then we resuspend it in a bit of media. And so now you have your cells resuspended in this media, and you can take a sample and count how many cells there are, and also get an idea of the viability of your cells, what percent of them are live, what percent are viable. So basically, Traditionally, cell counting is done with this like a hemocytometer. So it's basically this glass plate, and it has these typically two little things. You see there's like this little wedge here, and then there's like a glass plate that you put a cover slide over it, and now you have this little channel where you can stick cells into, and the cells, when you stick it in there, it'll like kind of like get sucked up. I mean, it's just through like capillary action and stuff, basically, because the water is like attaching and it just like gets pulled in. It's not like there's anything electronic or that sort of thing pulling things in, but it'll just kind of go in. And then on this hemocytometer, there are these grids, and then you count the number of cells per each of these grids using like some sort of cell counter to help you keep track. And then you are able to count the number of cells and do the math and that sort of thing. I really hate using these things. This is me in grad school with, these are SF9 cells, so these are insect cells. But this is just an idea of what these things look like. These days, thankfully, automatic cell counters are often used. So a really common one is this Countess. They are not paying me to do this. I do not do any endorsements or sponsorships or anything like that. But this is just what we use, what a lot of people use. And so this is what it looks like. You have these plastic slides. Again, it's kind of like that hemocytometer where you stick a sample on one side and it gets sucked in. And then these go into this machine, and the machine is actually going to do the counting for you, which is really nice. So you can see here, basically on the left, this is just the without it trying to calculate anything. You can kind of see that there are some cells that look like they have some they're darker. That's the ones that are trypan blue stained. But you can see that it's kind of hard to tell. And so this is one of the reasons, well, I'll get into later, where there are also like some people use alternative methods, and why there are some like caveats when using this technique, and it's not perfect. But this machine, which is really helpful, is able to actually detect which ones are dead and which ones are alive without you having to try to squint and figure it out yourself. And so this, when you put your sample in the machine, basically it's going to tell you the concentration. And so the concentration is, so here it's like 3.62 times 10 to the 6 per mil. So basically that's saying there's about 3.6 million cells per milliliter. But only 97% of them are alive. And so now it has this corrected, so that you can see that now there's 3.5 times 10 to the 6 viable per mil. And so this was the number that I would then use to do the calculations to figure out how much I need to add to my to whatever like volume I want in order to figure out how much to seed in my next experiment. And typically I'm doing these on a paper towel because I'm trying to keep things sterile in the TC room, but then transferring them to my notes and my nice notebook and my electronic notes later. But just an example where you can see that you can calculate how much you have, what concentration you would need to dilute it to in order to get whatever seeding density that you want. And I'll post a link to a helpful useful numbers for cell culture guide for trying to determine how much to seed various plates, and also it depends on when you want to next split them, or like harvest them or whatever you want to do next with them. Like the lower you seed it at, the longer you get to wait before you use them, or the longer you have to wait before you use them, depending on your point of view. Okay, just a little technical note. So when you're using the Trypan blue, another thing that's nice about this is you can see that it's Trypan blue dilution corrected. So the Trypan blue, when we use it, is typically like 0.4%. And it's normally like 2x. So basically you add one volume of it per volume of your cells. And so what I'll normally do is I'll add 10 microliters of my cells to 10 microliters of this Trypan blue, up, down, up, down, up, down, and then into the slide. And then it will take into account that dilution and when it tells you the number, so you don't need to do anything to this number. If you're using a hemocytometer, though, you do need to remember that you need to take this dilution factor into account, because what you're counting, the actual cell solution itself, was actually twice as concentrated, but then you diluted it with the Trypan blue. So keep that in mind if you're using one of the traditional hemocytometers, that your actual cell concentration when you're using the Trypan blue is going to be twice what you've measured here. Okay. Some more technical notes. So you want to work quickly when you're using the Trypan blue, so measure it within a couple minutes, because you can have artifacts form if you treat them for too long. So here are a couple of papers. I should just mention that, like, I could only find a couple papers that actually talk about these artifacts and various things, and these papers are coming from companies that are trying to sell you things that's like alternatives to Trypan blue. So basically, Trypan blue is typically good enough for the cell culture work. If people are doing things with like fluorescent sorting or that sort of thing, they're using some sort of fluorescence machine already, then there are better alternatives for determining cell viability. I'll talk about that in a minute. But bottom line, like when you're just doing cell culture, if you just need a basic idea of like what the relative concentration of the cells are, what the relative viability of these cells are, then the Trypan blue is typically like perfectly fine. It's only if you need more sensitive things, if you need more exact things, that you would potentially go to a different method, like something fluorescent. But then you're going to need special equipment, whereas here you just need the microscope. You don't need that Countess machine, you just need the microscope, and that will do. But there are things to keep in mind, though, when you're using it. One is that it can kill cells that weren't dead already. So we say that the live cells can't take in the dye. Well, they can't take it in, at least initially, but if you fill them with dye for long enough, some of it's going to get in and cause and kill the cells. So when this dye gets into cells, it can bind to proteins, it can inhibit all sorts of stuff and cause the cell to die. It can also cause cells that were dead to disappear. So basically, they get really faint or they even burst, so you undercount them. So what would happen? So the one, like, if it's killing cells, so over here you can see that the amount of the dark cells, so the stained cells, is increasing over time. So here, this TB, this is the Trypan blue. This PI, this is propidium iodide, which is a fluorescent alternative that's going to stain DNA and RNA of damaged cells. So you can see that more cells are dying over time. With here, and then acridin orange, this is going to get into all cells and stain the DNA. So this would be like all cells, and then this would be dead cells. And you can see that the amount of these dead cells is increasing over time. And you also see that there's some of these like things that are like these weird diffuse bodies, which they say like you have basically I think that's where they're pointing at arrows and stuff, is that these cells that are dead are kind of getting like burst and diffuse, and then they're kind of disappearing and making it hard to see. So when that happens, so when the cells die that weren't dead already, so you have these cells there that were living in the beginning, and now over time, these cells are dying. So now if you go and measure it later in time, there's going to look like there's more dead cells. And so you're going to underestimate the viability. So you're going to think that the cells are less healthy than they are. But if you have the other effects where the cells that were dead are now kind of like disappearing from view, now that can lead you to undercount the dead ones, which means you're going to think that you overestimate the viability because you think the cells are more alive than they actually were. And then again, it's like can affect the morphology. Like we saw, the cells were kind of like getting weird and diffuse. It's also not perfect at showing dead versus live cells. So some cells with like compromised membranes might still stain a little blue, but be salvageable, whereas cells with intact membranes will be unstained, but they might be non-viable because there's more ways to die than just having like your problems with your membrane, or maybe those problems aren't apparent yet at various things. There's a lot of things that can happen in a cell, a lot of ways to not be viable, but still not taking that dye. But hopefully they'll cancel out, okay, the two sides of the coin for both of these cases, and therefore be good enough. It's also not super sensitive, and it's going to stain different cells a different amount. So like if only a little bit of dye gets in, the cells are going to be a lighter blue, and if a lot gets in, they'll be a darker blue. As we saw when we were looking at the Countess screen or whatever, like it could be really hard to tell whether cells are actually dead or alive. So you can see here the ones that, this is basically, some of these are dead and some of these are alive, but it can be really hard to tell. With the fluorescence methods, then you can tell more easily, but then again, you need to have the fluorescence machine. But yeah. Okay, yeah. But as I said, it's quick, easy, typically good enough. So it's very, very commonly used. But there are alternatives that require more special equipment. There are ones like if you're already doing an experiment where you're using some sort of fluorescent sorting or something like that, then there are things you could do, like staining with the AO, which we look like, DNA staining, like DAPI. Yeah. Or they have some sort of like commercial things that you could use, like in the lab, in the TC room. But again, the Trypan blue is normally plenty good. Yes. So other things I'll just link to in the text. And then there are also techniques to determine viability based on the cells actually doing things. So based on metabolism. So metabolism is the making and breaking of molecules. So you can measure if the cells are using a lot of ATP. So are they breaking down ATP? Are they making lots of products we can measure? So there are various metabolic assays that can be used to test the metabolic activity of the cells, often ATP based. But again, these are going to be more complicated things than just sticking a little dye on a sample of your cells. So final fun note, the trypanosome. So final fun note, the name the Trypan dye that comes because it can kill trypanosomes, which are these types of parasites, these like protozoan parasites. So if, as we saw before, if this Trypan dye gets into cells, if those cells were living or in the beginning, well, now those cells are going to get killed by this dye. This dye can bind to a bunch of proteins, it can inhibit them, various problems. So it has a hard time getting into our cells, as we've seen, but it gets sucked into trypanosome cells through this process called endocytosis. So basically endocytosis is where there's like the cell, it kind of sucks in a part of itself. So it's like if you were to bite in part of your cheek, like, and then, yeah, so it's like swallowing part of its membrane in this like little like pocket. It sucks it in. When it sucks it in, it makes this little bubble, this little like lipid surrounded, membrane surrounded bubble called the endosome, which can then like things can get released from it inside of the cell. What was outside of the cell though, what was on the surface of the cell now gets swallowed and it gets inside the cell. If it can get out of that endosome, it's going to get into the cell, its interior. The trypan, this dye can bind to like proteins on the surface of the trypanosome. So if the trypanosome then endocytoses, it can take in the dye. And these trypanosomes do a lot of endocytosis because they have this, they think they're really clever, and then they get tricked by their cleverness. But normally this cleverness works. Basically, there are antibodies that can recognize the sugar, like a sugar or a glycoprotein, so like a sugar-coated protein on the surface of these trypanosomes. And if these antibodies bind to that, then they can bring in, like, call in the immune system, and the immune system can destroy the trypanosome. To avoid that, the trypanosome says, oh, there's like an antibody bound to me, let's suck it in, and haha, now the immune system doesn't know there was an antibody bound there.
But when you have the trypan blue, now it's going to suck in the trypan blue as well, and then it gets killed. So yeah, I didn't know about this until today, but here are a couple. I'll link to these. There's a couple of really cool articles. There's actually a drug called Sumarin, which is based. It's like an analog of the and of trypan blue that's actually used to treat trypanosomiasis. So pretty cool stuff.